Saurabh's classes. This is Ashmita. We are going to solve certain questions based on biotechnology question paper from this year, IIT JAM 2023. I have chosen 5 to 6 questions for today's class. Let's start with question 6. I don't know whether it will match with the set that you might have got because the sets were different in the question papers. Restriction enzymes that recognize the same nucleotide sequence but cleave at different positions are called. Now, these are called isochizomers. Okay, so go with option C. Talking about restriction enzymes, let's discuss a bit about this. Restriction enzymes are enzymes that cleave DNA at specific nucleotide sequences. And isochizomers isochizomers, they are restriction enzymes, just as it is mentioned in the questions. These are restriction enzymes that recognize the same nucleotide sequence, okay, but cleave at different positions. That's the only different thing. Um, this property can be useful in molecular biology applications such as in DNA mapping or in cloning, where the use of different isochizomers can provide a means of generating different DNA fragments with the same sequence. Okay, so that is what isochizomers are. Let's discuss about the other options also. Now, we will be discussing about the wrong options also because we need to discuss in details. Let's start with the very first one, that is heterohypicomers. Okay, heterohypicomers and the last option, that is neochizomers. These are not terms which are commonly used to describe restriction enzymes. So that is why I have not considered these as the correct options. Heterohypicomers, these may refer to complexes of different subunits or proteins that recognize different sequences. While neochizomers, these are or these may refer to the rather modified versions of the known restriction enzymes that may cleave at different sites. However, these terms are not specifically like used to describe restriction enzymes that recognize the same sequence. Okay, that is why I have not considered these options. Next, let's move over to the next question. So we have already got the correct answer for the previous one. Let's move over to the next question. Which one of the following does not belong to the freshwater ecosystem? It will obviously be estuary. The others are a part of freshwater ecosystem. So estuaries are basically coastal bodies of water. We all know about that. Coastal bodies of water that are influenced by both fresh waters and salt water from the oceans. As such, they are not considered part of the freshwater ecosystem, which is defined as, now what is freshwater? Freshwater can be defined as bodies of water that contain less than 1% of salt concentration. Less than 1% of salt concentration. Now talking about the other options which are not correct according to us. Let's talk about lentic and lotic ecosystems. Okay, what are these actually? Lentic and lotic ecosystems, or yeah, ecosystems only, are both types of freshwater ecosystems. Okay, and they have asked which is not belonging to this freshwater ecosystem. So that is why I have chosen estuary. Lentic and lotic are a part of freshwater ecosystems, and lentic ecosystems, if I talk about this individually, lentic ecosystems are still or standing bodies of water, okay, such as we can include lakes. These are not flowing, these are standing bodies of water, like lakes, ponds, wetlands. Hopefully you all never saw these water bodies or water in them to move from one place to another. It's not like that. They do not even drain anywhere. Anyway, while lotic ecosystems are flowing bodies. That's the difference between lentic and lotic. These are flowing bodies of water, such as rivers and streams. Okay, rivers, streams. Now, wetlands. Why is wetland not considered to be the freshwater a part of the freshwater ecosystem? Because wetland is itself an example of freshwater ecosystem. Okay, as they are areas of land that are saturated with water, and they support a unique community of plants and animals that are adapted to living in wet conditions. That is why it is like from itself it is known as wet land. Okay, let's move over to the next question. That is, which of the following is transcribed by RNA polymerase 3 in eukaryotes? Okay. Here, your correct answer would be tRNA is transcribed by RNA polymerase 3 in eukaryotes. Okay. Now, RNA polymerase 3. Let's talk about this one in a little bit detailed manner. RNA polymerase 3 is responsible for transcribing a variety of non-coding RNAs, including we can include transfer RNAs. Okay, that is tRNA. That is there in the option we have just chosen. Then small nuclear RNA. Okay, and some small nucleolar RNAs. Okay, and tRNA is responsible for carrying the amino acids to the ribosome during protein synthesis, and is therefore like essential for the translation of mRNA into proteins. Okay, that was all about the tRNA. Let's discuss about 18S RNA and 28S RNA. Now these two are transcribed by RNA polymerase. Okay, these are transcribed by RNA polymerase, polymerase 1, which is responsible for transcribing the large ribosomal RNA, that is the rRNA we call it, genes that make up the majority of the ribosome. Now talking about the miRNA. Now miRNA, or that whatever we call it, the microRNA, is transcribed by RNA polymerase 2. Okay, here it's transcribed by RNA polymerase 2, or the second. Now this is responsible for transcribing the protein coding genes as well as some non-coding RNAs. Okay, that's the work. And we can also say that miRNA also plays a role in gene regulation. Okay, by you can say by binding to complementary sequences on messenger RNA or mRNA, and also inhibiting the translation or causing the degradation of the mRNA. Okay. All right. That was it for today's class. If you have further questions, you can comment down below. And if you want to join our classes for better preparation and discussions and doubt clearing sessions, then you can definitely go to the description box below. There you will get all the details regarding our classes. Thank you for watching and all the best for your exams.